Let's talk a bit about biology. Let's start off by breaking the word into its components. Bio, the root, and logi, the suffix, mean life and study of, respectively. So in short, biology is the study of life. Now, what is biology to its entirety? Biology is the study of organisms and the impact of microscopic organisms, such as cells, on macroscopic organisms, such as animals. Now let's talk about experimentation and experimental design. This is how biologists correctly carry out their experiments. The components of a lab must include the investigation question. This question is what is trying to be investigated or what is trying to be found via this experiment. It's more or less the purpose of the experiment, and it should technically guide the whole experiment by itself. Next, we have the hypothesis. The hypothesis is a prediction based upon background knowledge of this experiment in which they're trying to carry out. It's always a good idea to have some background information about the experiment that's being carried out so that a valid hypothesis can be created. In this sense, it does not always have to be correct. It can be proven wrong, but it should be in the scope of the experiment. So, what is a good hypothesis? Let's talk a bit about that. A good hypothesis should include the independent variable and how it affects the dependent variable. So let's start by talking about what an independent variable is. The independent variable is the variable that is being manipulated or changed throughout the scope of this experiment. The dependent variable is the effect of the manipulation by the independent variable. So you can think of this more or less as the cause and effect, the independent variable being the cause and the dependent variable being the effect. Now, to write a good hypothesis, you need to create an if-then statement which encompasses the independent variable and the dependent variable, explaining what might happen in this experiment. This experiment should either prove or disprove your hypothesis. That's really important. So the hypothesis cannot be a qualitative observation of some sort in which the experimenter cannot come to a conclusion of. It should be something that can be tested and proven with the quantitative data that is present. Now let's talk about what control variables are. So control variables are variables that reduce any other external factors into the experiment. These try to limit any other variables that might change the effect or the result of the experiment. Lastly, we have the control group. The control group is the group of objects or subjects that are being tested upon without the independent variable acting upon them. This is to make sure that the experimenter can compare their information or their final results with that of the control group. Let's start off by asking if increasing the temperature increases the rat's diet. So that would be the first step, making our investigation question. Next, we have our hypothesis. One might hypothesize that an increase in temperature increases the amount of food eaten by the rat. This is obviously measurable since we can easily measure the amount of food that was given to the rat or eaten by the rat. Now, let's identify the independent variable. The independent variable in this scenario is temperature. The dependent variable is the diet. So the diet is dependent, or how much it eats, and the temperature, I'm just going to abbreviate it, is the independent variable. Next, we have our control variables. So what are we going to keep constant so that it doesn't interrupt this experiment? Some of the control variables might be the same location, same breed of rat, and etc. So in this scenario, we might make the control group just regular without any temperature changes. So room temperature might be our control group. So control group rats will be at room temperature. Now, this will help us figure out if there was actually a change with the temperature affecting the rat's diet.